I can briefly introduce to the SQS instrument. SQS means small quantum systems. That means on this instrument we are studying mainly atoms, molecules, clusters, or small quantum particles. And what we study is the interaction of these particles with the intense FEL beam. And in general, there are three different types of large scientific topics which we try to cover. First one is really, let's say, to make use of the intensity of the high intensity we have here for the free electron laser. That means we are studying nonlinear processes, multi-photon excitation, multiple ionization of these species like the atoms and molecules. And it's particular that you have these samples in the gas phase, for example, atoms, then you can study these effects in a very fundamental level. You really don't have interaction with the environment. You have really only the sample, the single atom, which is, let's say, on the basis of the interaction from light with matter. The second topic is related to time-resolved experiment, where we make use of the short pulse duration of the free electron laser. And this is also mainly then in combination with a synchronized optical laser. What we are studying there is the dynamics of the molecular fragmentation, for example. That means you trigger with one photon the fragmentation of a molecule, and with the second photon you probe the individual steps in a time-resolved way, how this, let's say, system, this excited system, is evolving. And the third topics are, let's say, related to the larger systems, clusters, nanoparticles, or even larger biomolecules, where we do imaging experiments, where we try to get information about the structure of these large samples. This is similar to what you also have seen at other instruments. For us here, because SQS is the instrument at the soft X-ray regime, we have, let's say, compared to the other hard X-ray instruments, a much higher cross-section for the scattering, but on the other hand, okay, will not reach the final spatial resolution due to the wavelengths which we have here available. Okay, at the entrance of our hutch, we have a diagnostic section, which mainly consists of two components. One is a gas monitor detector, which allows us to measure at the entrance of the hutch the intensity of the incoming FEA radiation. Second is an imager, which allows us to define precisely the position of the incoming beam. This is particularly important because you want to bring the beam as an optimal position to our optics, focusing optics, which is the next component in the beamline layout. And these are two highly polished bendable KB mirrors, meaning that we can change the focus size and the focus position. But it allows us, on the other hand, to have a very tiny focus. And the tiny focus means for us a focus with a size of the order of 1 micron. And this allows us in particular to drive these nonlinear processes, because you have to bring a lot of photons in a very small volume. The chambers, I mean, what is the next point are the experimental chambers for the different type of experiment, which I have told you about at the beginning. There we have three different types of experimental chambers. One is the so-called AQS chamber for atomic-like quantum systems. As I said, is for small quantum systems like atoms and molecules. The main focus of this chamber is really to study these nonlinear processes and the time-resolved studies. As detectors or analyzers, what we have there available are mainly electron time of flight spectrometer, ion time of flight spectrometer, a velocity map imaging for getting the angular distribution of the electrons or fragment ions, and the last part is a magnetic bottle electron spectrometer, which allows us to really have a very efficient acceptance of electrons. That means you can really study very tiny processes, which gives you only a very small signal. The second experimental station is the so-called reaction microscope, which is at the moment not installed at the beamline. This is also dedicated to experiments on small systems like small molecules and atoms. And the main advantage of this equipment is that you can measure electrons and ions in coincidence. And you do it in such a way that you have the full information about the emission angles and also about the kinetic energy for electrons and ions at the same time. And this allows you really to do the complete characterization of the process which one wants to investigate. The third experimental station which we have available here for users at the SQS instrument is the so-called NQS station. NQS means nano-sized quantum systems, and it's dedicated, as its name says, to larger systems, to clusters, to nanoparticles, and also of biomolecules. This station is at the moment in a sort of parking position in the middle of the experimental hall, so it's not here directly at the instrument. The main components of this stations are first the cluster source, which is on this side, which is really a dedicated one to produce these larger systems. The second are some ion time-of-flight detectors, which are used, for example, to look if there's any fragmentation processes produced by the interaction with the FAL beam. And the third and the main thing is really an imaging detector. This is a 1 megapixel detector called PNCCD, which really gives then the information about the size and the structure of these particles. And this is a dedicated detector which is working or optimized for the soft X-ray regime.